Hi guys, today we get to talk about meiosis and sexual reproduction. So it's a big day in science. So we've already talked about mitosis, and we talked about how mitosis is the process that eukaryotes use for growth and repair for the most part. There are some individuals who use it for asexual reproduction, but for the most part, we're looking at mitosis for growth and repair. And what you'll see is that you start off with the mother cell, there's growth signals, something needs to happen, growth or repair or something like that. And so then that mother cell will divide and you'll end up with two genetically identical daughter cells. So by genetically identical, that means they have the same chromosome number as the parent. The daughter cells both match the parent cell, and that's how it goes. Meiosis is a little unique, though, in that it's a special type of cell division that is used only for sexual reproduction. So you can end up, because it's such a huge, it's such a process that allows for a huge amount of diversity. You could have, just with sexual reproduction alone, you could have up to 70 trillion different genetic combinations by combining gametes. And so the whole process of meiosis is that it basically halves the chromosome number prior to fertilization. And so the parents are diploid, and the parents produce haploid gametes. So remember, diploid is both sets of chromosomes, and haploid is half of those chromosomes. So meiosis produces haploid gametes, and then fertilization brings those gametes together to form a diploid zygote. And then that zygote goes on to be the next diploid generation. Remember we talked about ploidy in the last chapter. We talked about how that refers to the chromosomes in the cell. Remember that haploid is where you'll see one set of chromosome, and if you're going to shorten that, you can write it as N. You'll see it as N often. And diploid refers to two sets of a chromosome, so or two times the number of chromosomes. So haploid and diploid terms refer to the chromosome amount, not the organism versus sex cell. So for diploid cells, chromosome numbers occur in pairs. So humans have 23 different types of chromosomes, and in each body cell or somatic cell, those chromosomes are represented in a diploid number because you have a chromosome from each parent. So because it's diploid, you have a chromosome from each parent, you'll see that chromosomes of the same type are said to be homologous chromosomes or homologs. So with homologous chromosomes, this basically means like this chromosome that you got from mom is very similar to this chromosome that you got from dad. Obviously the genes are different, but their centromeres are in the same place. They came from, one set came from dad, one set came from mom. They have similar banding patterns if you were to stain those chromosomes. And they have genes that control the same trait at the same position. And so that's what it means to be a homologous pair of chromosomes. One came from mom, one came from dad, but they have some similarities, and of course they're gonna have the genes that control the same trait at the same position. And each gene will occur in duplicate. You get mom's copy and dad's copy. So when you're looking at chromosomes, you'll always notice that anytime when we talk about them in lab or lecture, we talk about these chromosomes, which we learned from mitosis that these are visible now. These are sister chromatids, and they're visible when they're condensed. And so a karyotype is a map of all of the homologous chromosomes. So it shows you all these chromosomes arranged by size, shape, and banding pattern. Um, and so this is chromosomes at location 1 all the way down to chromosome 23. Numerically, you get 1 through 22, and that's because that 23rd chromosome is the thing that, these are all body cells. This is the chromosome that determines sex. So a gene is a part of DNA that contains information for a trait. So genes happen in duplicate because you get one from dad and one from mom. And you can have lots of variations and lots of forms of genes. And those are referred to as alleles. So if you see various forms of genes, you'll see it's an allele. Homologous copies of a gene may code for identical or different genetic information. So the copy from each parent may be different or similar. So we talked about how an allele is a variation of a gene. So an individual might be homozygous or heterozygous for a particular trait. If you're homozygous for the trait, you have the same allele from both parents. And so identical alleles for specific genes appear on both homologs. If you're heterozygous for the trait, the maternal allele differs from the corresponding paternal allele. And so that actually makes me think of a story. My daughter's dad has these like really long really long toes that are like the length of fingers, and mine are just like tall, smaller, so smaller, like that. Um, And so I used to laugh, like when we were getting ready to have our first child together, I was like, oh my gosh, what if that's like a really dominant trait? And what if, she inherits your toes, which I used to call tingers, just to give him a hard time. And so I used to wonder, well, okay, so maybe long toes is recessive trait, and maybe my short toes is dominant trait, so maybe she'll come out with one or the other. But I imagined I imagine that maybe she'll be heterozygous for that long toe trait because he's got long toes and I have short toes. And so if we both had long toes, we'd be homozygous, or if we both had really short toes, we'd be homozygous for that toe length. And that's very generic, and it's a silly way to look at genetics, but I always remember that funny idea. So meiosis itself, so remember the whole process of mitosis was growth and repair. The whole process of meiosis is sexual reproduction. And so the goal is to take a cell and make haploid cells. You don't want to make gametes that you're going to share and hope to fertilize with someone if they're already diploid, because you're going to make the wrong chromosome number of an individual. Like humans have 46 chromosomes, so we really want our gametes to have 23 and 23. So mathematically, we make one 46 chromosome individual. So the process of meiosis halves that chromosome number so that you can have your gametes with half the chromosome number. And so because you have to half the chromosome number, so you get haploid gametes, 2 nuclear divisions are required. So those nuclear divisions are not hard to remember. So there's meiosis 1, which reduces the chromosome number. There's a short interkinesis, which is kind of like interphase, but you don't double the chromosomes, obviously, because you're trying to get rid of some of them. You're trying to half it. And then there's meiosis 2, which divides out those chromosomes to all the gametes. So during meiosis 1, chromosomes are replicated prior to meiosis 1. Each chromosome consists of 2 identical sister chromatids. Homologs actually pair up in a process called synapsis. So this is kind of unique to meiosis 1 in that those homologous chromosomes pair up and line up at the metaphase plate. Like we see chromosomes line up at the metaphase plate in mitosis, but we're not seeing homologous pairs line up at the metaphase plate. So it looks a little different. If you can see, with mitosis, you've got your chromosomes lined up at the metaphase plate, but meiosis, you can see that there's those homologous pairs. You get some from mom, some from dad. Those are lined up when you see synapsis. So then each daughter cell receives one duplicated chromosome from each pair. So this shows you just an overview of meiosis. If you look, you start out with the parent cell that's diploid. This is still diploid. DNA was reproduced before, like during interphase, and then here's your reproduced sister chromatids. But also, if you look at the different coloration, there's also homologous pairs. So there's these guys. There's these guys from dad, these guys from mom. And so then those are going to divide. Those are going to, once the first nuclear division happens, half the homologs are going to one cell, half the homologs are going to the other cell. So some are going this way, some are going this way. So during meiosis 2, DNA actually doesn't get replicated, obviously, because you're trying to reduce the chromosome number, not increase the chromosome number, right? During meiosis 2, the sister chromatids separate and move to opposite poles. You'll end up with 4 daughter cells containing 1 daughter chromosome from each pair. So each chromosome consists of a single sister chromatid. Now those daughter cells are haploid. And so at the end of mitosis, you get 2 genetically identical daughter cells, but at the end of meiosis, you get 4 genetically unique daughter cells. This shows you the unique thing, again, about meiosis 2, which makes it a little bit different, is that after that first nuclear division, you can see that the sister chromatids are still attached at a centromere. And so you know your work here is not done because you got to separate these, right? You got to separate those centromeres so that they can be chromatids instead of sister chromatids. So with the process of meiosis, it actually has the ability to bring about genetic variation in 2 different ways. We know that just a simple joining of gametes, like joining of egg and sperm together, gives you like so many, almost limitless genetic possibilities. Just taking the genes from your family and joining them with the genes from your partner's family. However, in addition to that, there are other things that happen during this process that bring about variations in genes. So 1 is crossing over between homologous chromosomes, and 2 is independent assortment of homologous chromosomes. And we'll talk about those. So crossing over is basically exchanging genetic material between non-sister chromatids during meiosis. So once synapsis happens, those homologous chromosomes will stick together and align DNA of non-sister chromatids and allow crossing over to occur. And so then the homologs separate and are distributed to separate daughter cells. So that's what this looks like. You got your homologous pair. They do this little cute dancey dance, and then they exchange genetic information. So that gives one way of giving variation. So another way genetic variation is brought about is through independent assortment. So when you see those homologs line up at the metaphase plate, they separate randomly. Like there's no predetermined way. Like the mom homolog doesn't have to go one way where the dad doesn't have to go the other way. It's really random. And so that causes really mixed blocks of alleles that end up forming into gametes. So if you look, you could get you could get any of these 8 combinations just because you have this number of homologous pairs. So then fertilization on top of that brings a huge amount of variation. So you could make gametes, you could make eggs or sperm with like the difference possibilities, the genetic difference possibilities is this number here. Crossing over happens only once, but it happens more than that usually. Think about that. So obviously it's very significant that genetic variation is a thing. Individuals or groups that have asexual reproduction always produces identical clones. So when you have identical clones, if something were to happen within the environment, there's some change in the environment, identical clones have less of a chance of survival because there may not be an advantageous gene there. Now, for those of us who do live in a changing environment, a very changing environment, we do see genetic variability, and that's advantageous because some offspring may have a better chance of survival if there's an environmental change. So the phases of meiosis 1 and meiosis 2 are not that different from the phases of mitosis that we have already learned about. So the only difference is, of course, remember meiosis 1 is the first nuclear division, meiosis 2 is the second nuclear division. And it's really, so in meiosis 1, you'll see prophase 1, metaphase 1, anaphase 1, telophase 1, and then you'll see a little pause called interkinesis. And then you'll see in meiosis 2, you'll see prophase 2, metaphase 2, anaphase, telophase 2. So it's very similar to mitosis, only it happens twice. So we can divide that nucleus twice, and so we can get half the chromosomes in 4 gametes. So during prophase 1, it's a lot like prophase in mitosis. A spindle forms, nuclear envelope fragments, chromosomes are duplicated. Only in this portion, you'll see homologs line up side by side. It's synapsis, and sometimes people call the synapse homologs bivalents or tetrads. I like tetrads because I think of tetra and I think of 4 for me, so I usually say tetrads. So then at metaphase 1, homologous pairs are arranged at the metaphase plate. Those bivalents or tetrads are aligned independently of one another. And then anaphase 1, you'll see the homologous chromosomes of each tetrad or bivalent separate from one another, but note that the sister chromatids are still held together at this point by a centromere, so that's a big deal. They don't get separated. Then telophase 1, you'll see that you have daughter cells that have 1 duplicated chromosome from each homologous pair, and so you'll end up with 2 haploid daughter cells, but you're not done with your work. Even though the goal is to get haploid cells, you're not really done with your work because your sister chromatids are still stuck together at the centromere, and we need to divide again to separate those dudes. So everything looks great with meiosis 1, and life is going grand, right? Because you've separated, you've got 2 cells, only you're not done because your centromeres are still stuck there, holding those sister chromatids together. During meiosis 2, what happens is that mitosis happens in both of those haploid cells that we made, okay? So we'll see prophase 2, metaphase 2, anaphase 2, telophase 2, and it's exactly like mitosis, only it's happening in both of these cells at the same time. So prophase, metaphase, anaphase, telophase is happening here and here as well. So in anaphase 2, same kind of thing, centromere dissolving, telophase 2, and cytokinesis, you end up with 4 haploid daughter cells that are all genetically unique, which means they're all different. They have all different genes in them. This is an image that comes from your lab manual and your textbook both, and it shows you meiosis 1 in a plant and animal cell. Here's meiosis 2. So you eventually get those 4 genetically unique daughter cells. So it's kind of big to know. Whenever I give an exam, I really like to know that you know the difference between meiosis and mitosis. So of course meiosis requires 2 nuclear divisions, mitosis only requires 1. You'll see synapsis in meiosis, and you don't see that in mitosis. In meiosis, centromeres will survive anaphase 1, and that doesn't happen in mitosis. Meiosis halves the chromosome number, and mitosis preserves the chromosome number. Meiosis produces 4 daughter cells, where mitosis produces 2 genetically identical daughter cells. Meiosis is only used for sexual reproduction, whereas mitosis is used for growth and repair and asexual reproduction. And this is a visual comparison, which is really nice. So when we're talking about chromosome number or ploidy, bacteria are haploid. Mosses stay haploid most of their life cycle. Ferns and higher plants are diploid most of their life cycles. In social families of insects, bees, ants, wasps, the females are diploid while the males are haploid, and that encourages those social interactions. In flowering plants, you have a diploid male that produces haploid gametes, and that haploid gametes, which is pollen, gets released, and haploid sperm is released once it releases the female structure, and then of course a fertilized ovary will then develop into a fruit. So humans are diploid and produce haploid gametes. The products of meiosis are always gametes, and that's the only haploid part of the life cycle. And so gametogenesis occurs in the sex organs of males and females. With spermatogenesis, that's where you'll see the production of sperm. And all 4 of those cells that are created at the end of meiosis become active sperm. Oogenesis is the production of eggs, so only 1 of the 4 nuclei receives the majority of the cytoplasm, and only 1 of those 4 cells actually becomes the egg or the ovum. The other ones go away as polar bodies. So sperm and egg are produced by the process of meiosis. Those sperm and egg fuse at fertilization and form a zygote, and all growth that occurs after fertilization is all due to mitosis. So because of that, because of mitosis, each somatic cell or body cell in the body has the same number of chromosomes as the zygote, and it has the same makeup. So this shows you the life cycle of humans, where you start out as a diploid zygote, and many mitoses later you become a baby, then you become an adult, and then you produce as an adult haploid gametes that would fuse together at fertilization. This shows you spermatogenesis in males, and of course oogenesis in females. So males actually have testes in a scrotum that contain stem cells called spermatogonia. Those spermatogonia provide primary spermatocytes that undergo spermatogenesis. Those primary spermatocytes have 46 chromosomes, okay? So I'm gonna draw this. Okay, so you start out with a diploid primary spermatocyte, right? And after meiosis 1, you end up with 2. I'll show you first. Here's my one individual primary spermatocyte that's diploid. After meiosis 1, you end up with 2 secondary spermatocytes, which is great. They're haploid, but they're not really haploid yet, because remember, at the end of meiosis 1, you have those 2 daughter cells that are haploid, but the sister chromatids are still stuck together, so you got to do another division so you can separate those sister chromatids. So meiosis 2 gives you 4 spermatids that are all haploid. So if you see my spermatogenesis drawing, you've got your primary spermatocytes up, let's see here, primary spermatocytes, then you got your haploid secondary spermatocytes, and then your totally haploid spermatids, and each of these have 23 daughter chromosomes, and then they'll differentiate into your 4 little viable sperm cells. So there's your 4 little happy viable sperm cells. So spermatogenesis is super straightforward, because you're like, okay, meiosis 1, bam, meiosis 2, bam, bam, bam. Like, it is what it is. Oogenesis, though, gets a little bit more complex, because with oogenesis, females have ovaries in the pelvic cavity that produce stem cell called oogonia. And that actually happens. Females produce primary oocytes with 46 chromosomes during fetal development. Okay, but oogenesis begins, but it actually gets halted until puberty. So you have your primary oocytes while you're developing in your mom's stomach, which is super crazy. Like, when you think about that, you're making your gametes while you're a fetus, while your mom is growing you. So your mom is helping to grow their own grandchildren. What? It's crazy. So, let's do some oogenesis drawings here. So you've got, this gets really hairy, but it's interesting. So you've got your primary oocytes, which is your primary oocyte there, which is diploid. Meiosis 1 happens. You get 2 haploid cells with 23 chromosomes, right? But only one of those haploid cells gets all the cytoplasm. One of those actually just is a polar body, and it's much smaller, right? So you've got your secondary oocyte that gets all the gooey cytoplasm, and you got your little polar body over here that doesn't get anything. And that polar body may disintegrate, or it may go on to divide again. So the secondary oocyte begins meiosis 2, but it actually stops at metaphase 2, which is, this is an independent and very like women-powered oriented kind of individual, this is trippy to me because, so that secondary oocyte it leaves the oviduct, or it leaves the ovary at ovulation. It bursts out of the ovary, but remember, it stopped at meiosis 2, so you don't have a fully formed oocyte yet. That secondary oocyte bursts from the ovary, enters the oviduct, and checks around. Any sperm waiting? No? Yes? So if sperm is present, so remember your oocyte is stuck at metaphase 2 during meiosis 2. So if sperm is present, then that sperm will actually trigger the rest of meiosis 2 to happen, and another polar body forms. And after oogenesis and the entrance of sperm, there is one oocyte and 2 to 3 polar bodies, which, if there's no sperm present in the oocyte, then or in the fallopian tubes or the oviduct, then it'll just go on to disintegrate. Okay, so that's what's trippy to me. So the oocyte does not complete the whole process of meiosis unless sperm is present to trigger that. So we can't complete meiosis on our own. We are required to have a sperm cell to complete that process. So there are instances where sometimes there are changes in chromosome number and chromosome structure. So when you see euploidy, that's the full number of chromosomes in a species. Aneuploidy happens when there's a change in the chromosome number, and that happens as a result of disjunction or some sort of faulty happening when gametogenesis or meiosis is happening. So when you see monosomy, there's only one particular type of chromosome. When you see trisomy, you have 3 of a particular type of chromosome. And there are lots of genes that interact across chromosomes, and the differences in this chromosome number can have drastic effects. So this shows you non-disjunction when you see a pair of homologous chromosomes and something happens and they just don't divide right. And then there's all kinds of things that can happen as a result of that. Trisomy happens when an individual has 3 of a particular type of chromosome. So in humans, trisomies can result in a viable offspring in birth. And the most common autosomal trisomy that we see is trisomy 21 or Down syndrome. So of course the chances of disjunction when gametogenesis or meiosis is happening, that chance of it happening and having some sort of issue increases with age, and it increases with the age of stored oocytes. And so that's why they say women over 35 who do have babies have an increased likelihood of having a trisomy happen, having something happen during meiosis or gametogenesis that causes some chromosomal imbalance there. And this shows you trisomy 21 and what it looks like. There can be issues when you see changes in the sex chromosome as well. So you can see issues when you get too many or too few X or Y chromosomes. Because you can see non-disjunction during oogenesis or spermatogenesis as well. So when you see that, you can have an individual with an X and then a nothing with a single X chromosome, and that's when you see Turner syndrome, or XXY when you see Klinefelter syndrome. You'll see you'll see underdeveloped testes and prostate, some breast overdevelopment, long arms, hands, near average intelligence unless you get more X's. You can see mutations or changes in chromosome structure um, that happens either maybe during meiosis or even environmental agents can impact chromosome structure. So if you have a deletion, you have one or both ends of a chromosome break off. You have a duplication, you can have the presence of more than one chromosomal segment. You can have a translocation where one chromosome segment moves to a non-homologous chromosome, which is crazy. An inversion. So the SRY gene it plays a huge role. It's a gene located on the Y chromosome, and it contains information for proteins that start testis development and stop female gonads from forming an embryo. That gene can be deleted, and that results in an XY individual with no testes determining factor. The individual will have XY chromosomes, but develop a uterus, uterine tubes, ovaries, subsequent hormones. And the gene can translate to an X chromosome, but an individual may be born XY or XS, but with SRY on the X. It's a huge deal, and I suggest you click on the link for this video so you can watch it. And I do have a really interesting case study regarding the SRY gene that we'll try to watch. We'll try to take a part in the SRY gene study if we get to it, because it really goes with our reproduction chapter and reproduction unit. It's really good. So then we get into, you have that issue with SRY, like you can be chromosomally male, but then have that faulty SRY gene and you don't actually develop those male genitalia, you develop female genitalia, and that can be it can be a big issue. But then there's also other considerations at what makes an individual male or female. So there are lots of situations where that doesn't apply. There's infertility, there's issues with aging, intersex individuals, there's all kinds of situations, and it's such a broad spectrum. It is not a black and white A, B, C, D thing. It is a spectrum. It's a spectrum of understanding. And so male and female can, in broad terms, denote sex organs, chromosomes, or gender independently. It's really a big, huge understanding. So sometimes the XXXY internal anatomy doesn't match the external anatomy. That happens. Sex organs and chromosomes and gender may not align. There's all kinds of interesting things. So individual anatomy and physiology determines sex. The presence of SRY gene, early environmental and developmental factors, gene expression, that kind of thing, they all play a role in sex determination. Most of the times we don't walk around going, you know what, I've seen my sex chromosomes and I know I'm an XX or I know I'm an XY. And there are lots of situations where intersex individuals are born with various forms of sexual anatomy that don't fit just male or female. So gender is generally masculine or feminine expressions of an individual, and it doesn't necessarily apply to sex organs that an individual has. So expression of gender can change with societal norms, definitely like clothing styles from the 17th century versus the 20th century. And gender expression is behavioral and not genetically encoded per se. And humans are inclined to mimic social norms to fit in, but that's not always the case. That's not always how it should be. There are individuals who are transgender individuals who have sex organs at birth that don't necessarily align with personality and personal identity. Just like sex determination, there's a lot of factors that affect how you feel inside. And cisgender individuals have sex organs that do align with um, your identification. Textbook definitions or more personal perceptions can be more strict than what happens in nature. Life doesn't read textbooks. Um, not every plant with leaves of 3 is a poison ivy, that kind of thing. So individuals of a population have variation. Humans are very complex. We're all very different. Um, Everyone has different experiences, and biology actually allows us to understand tons of amounts of variation can exist. Biology does not support bigotry.